This is Kanika Gupta, your TA for the session, and today is the session for week 11. I hope you guys have listened the lecture that was already there on the site. Let's go through them once and then we'll discuss the questions from the assignment only. And if you have any doubts, we can discuss them also. Okay, so let's start. So, in the last week's lecture, sir discussed about the genetic disorders. Okay, so genetic disorders means disorders related to genes, related to genome. So those are called as the what? Genetic disorders, which affect the which are because of some kind of problem in the genes. It's not like कि आपका कोई एंजाइम कम या ज्यादा हो गया, बट इट इज लाइक कि उसकी जीन ही जो है वो या तो म्यूटेट हो गई है या इट इज प्रोड्यूसिंग सम फॉल्टी प्रोटीन्स. Okay, so that are called as the genetic disorders. uh, then the different type of reasons for different kind of genetic disorders. For example, HIV-1 infection, that is the responsible mutated gene is chemokine receptor, and the target cells for editing is the CD4 positive T cells. So basically, when we are talking about genetic disorder, so the treatment to these kind of problems is the genome editing that has been used from last many years. That the genome editing is the only treatment that was there for the genetic disorders. So if will talk about the HIV-1 infection, so the responsible mutated gene will be CCR5. The target cell that will be edited for this is CD4 positive T cells, and the delivery vehicle will be either adenovirus or mRNA. Same goes for leukemia. Leukemia is another name for blood cancer. So for this, the responsible mutated gene is CD123 TRAC, that is T cell receptor alpha chain, and the target cells for editing is the chimeric antigen receptor T cells, CAR T cells basically, and the delivery vehicle is again mRNA here. Same goes for B thalassemia, sickle cell anemia, or Parkinson disease. So there are many kind of genetic disorders, and according to which cell is affected the most, or which cell can help us in recovering the defect that has been happened due to this genetic disorder, is the one which is used for the editing. Okay. The general approach for treating genetic disorder is, as I told you, collecting the desired cell type to edit. Because, as I told you, we need those cells upon the editing of which we can get a short result that after editing will get some. Just a will get some way to overcome the repercussions of the disease. So in that case, we'll collect the desired cell type to edit. Then the genetic editing tools will be used to edit the genome. Wherever the faulty gene is present, it will be edited. Then the packaging and delivery using the vectors. Then the growing cells were growing the transformed cells in which the edited genome has entered, and then transplant these cells in via injection into the affected human. So this is a general approach using the genome editing for treatment of this kind of genetic disorders. Okay, so if we'll go into the different types, first is the recombination, which is the core of genetic engineering. So, recombination is the genetic process involving the breaking and recombining of the DNA segments, resulting in the creation of a new combination of alleles. This mechanism operates at the gene level, fostering genetic diversity that mirrors variation in DNA sequences among organisms. So, recombination, if you some of you are from the biological background, so you must have heard this term when you were studying about the cell cycle in the meiosis part, that recombination, how recombination happens. So this is the same thing. Now the recombination can be homologous, non-homologous. Sorry. It can be homologous, it can be non-homologous. If it is homologous, it can be bacterial, it can be eukaryotic. Non-homologous has further many types. So first we'll talk about the homologous recombination. So homologous recombination is a genetic recombination process characterized by the exchange of genetic information between two closely related or identical molecules. Basically the sister chromatids here we are talking about. Sorry, not sister chromatids, the replicated form. The one chromosome is there, and you have a replicated form, the duplicated chromosome of that. The You will say the like one copy we get from mother, we one copy we get from father. So the one which we get from mother and from the father, for the exact chromosome, they both will recombine. They both will cross over and exchange some part of the chromosome. So that will be called as the homologous recombination. Okay, so it is a genetic recombination process characterized by the exchange of genetic information between two closely related or identical molecules of the nucleic acid, and these molecules can be either double stranded or single stranded, and they are typically composed of DNA in cellular organisms. Although in viruses, RNA may also be involved in the recombination process. Mostly it is involved with the repair mechanism and meiosis of the prokaryotes and eukaryotes. Okay, so this kind of thing happens in case of the meiosis. When we are talking about meiosis, the process is called as the crossing over, and after the exchange, some part of paternal chromosome will go to the maternal and some part of maternal will come to the paternal chromosome. And after the exchange, then both the chromosomes separate, so it is called as the homologous recombination or recombinant chromatids. Okay, so this is what is homologous recombination, basically the crossing over that occurs in case of the meiosis. Okay, homologous recombination, as I told you, is can be prokaryotic, can be eukaryotic. So if it is prokaryotic, it is further of two types, rec BCD pathway or rec F pathway. If it is eukaryotic, it can be DSBR or SDSR. Okay, so let's see in detail what is rec BCD. So we are actually not going into the detail of the process. So rec BCD is actually three units that come together to form this rec BCD complex as a whole, which binds to blunt or nearly blunt end of a double stranded DNA break wherever DNA break has happened. So basically this is a kind of the repair mechanism for DNA. Rec B and Rec D helicases work together to unzip the DNA duplex, and Rec B's nuclease domain cuts the emerging single strand. So basically, as I told you, it is a repair mechanism. It will see if any missing nucleotide is there. They'll help to put the correct nucleotides there, and if break has happened, a nick is formed, then they'll work accordingly. So basically we are again not going into the detail, but this is a repair mechanism for DNA. Okay. In the rec F pathway of homologous recombination, bacteria employ a repair mechanism for single strand gaps in DNA. Okay. When mutation inactivates the rec BCD pathway and additional mutations disable the SBC, CD, and exol nucleases, the rec F pathway can also repair DNA double stranded breaks. So basically, rec BCD is for the double stranded, rec F pathway is for single stranded. But rec F pathway can also work for double stranded breaks if rec BCD pathway is somehow mutated or inactivated, or some kind of a problem is there in the repair mechanism. Then in that case, rec F pathway is activated, which can repair both the single stranded break also and the double stranded break also. Okay, we are again not going into the details of the process. I hope you guys have heard, you guys have learned and understood these processes through the lectures. Then the when we are talking about the eukaryotic homologous recombination for repair. So here what happens is ATM is the protein which recognizes the double-stranded break, phosphorylates, recognizes the DSB recognizes the break, sorry, and DSB phosphorylates the H2AX and facilitates MDC1 binding. So basically, again, there are many downstream proteins that are involved in repair mechanism. One, there will be some for recognition, some will be there to activate the further process, some will be there to provide the raw material, and this is how the whole process will go on. Okay, so this is how these processes will go on. This is again a repair mechanism which is followed in case of eukaryotes whenever there is some kind of a break on DNA. Okay, two proposed pathways for eukaryotic homologous recombination mediated repair, that can be classical double stranded break repair or synthesis dependent strand annealing. ठीक है 2 मिनट I don't think this was the second name. Mm. Let's go for the double stranded break repair. So 3 dash ants invade an intact homologous template. Formation of a double holiday junction, DHJs they are called as. Okay, so they will form a double holiday junctions, and the junctions will result in the crossover or non crossover. So basically a new strand will try to invade the other double strand which is not affected, so that the affected area can be copied from there, and then the second strand of that copied thing can be just again someone entered the meeting, then that can be used to repair the break portion, the portion which has suffered. Okay, so the key factors and their role in eukaryotic homologous recombinations are the mRNA complex, which is used for the initial stabilization of a double stranded break. ATM, ataxia telangiectasia mutated, whatever, sorry, recognizes DSB. It will recognize the double stranded break as we have also discussed before. So BRCA1 and BRCA2 are the breast cancer associated genes. These are the checkpoint activators and also the DNA repair mechanism activators. Okay, so CTIP is the C terminal binding protein, interacting protein, interacts with BRCA1 and dephosphorylates 53BP1. RPA is the replication protein A which stabilizes the single stranded DNA. Otherwise, it is not the normal nature of a eukaryotic DNA to be single stranded. So, it must be stabilized so that whenever the repair mechanism is going on, the other strand is not affected. Okay. H2AX is a H2A histone family member X protein. It helps in the recruitment and accumulation of the DNA repair proteins. And last but not the least, RAD51, the DNA repair protein, which forms the filament on the DNA strand, basically the actual repair protein. Okay? So if we'll go for the summary of homologous recombination in both the organisms, so the introduction of the double stranded break, it can be due to SPO11 in meiosis and HO mating type. Processing REC BCD or REC F दोनों तरह के जो complexes हैं that can help in the processing and the repair mechanism. The pairing and the strand exchanges, REC A is involved. Branch migration, RUV AB complex is there, and resolution of the holiday junction, RUV-C protein. Where for the branch migration in eukaryotic, no proteins is known, and the resolution of the holiday junction, many proteins are involved, WRN, BLM, RED 51C, and so. So this is just to recall you key which proteins are involved where. Then comes the non-homologous recombination. So recombination involves the physical exchange of DNA segments between chromosomes or DNA molecules. When this exchange occurs between stretches of DNA with no extensive sequence homology, it is termed as non-homologous recombination. ठीक है. So there the recombination was happening between similar kind of chromosomes. Here there is no extensive sequence homology is there. It is termed as non-homologous recombination. Unlike homologous recombination, it does not require a double strand break in the DNA for initiation. It's not a mandatory condition for non-homologous recombination. It is also relatively less precise and error prone because यहाँ पे homology जो है that is not much. So obviously the chances of error is higher in case of the non-homologous recombination repair mechanism because it does not have some template by copying, by seeing which it can copy the exact sequence and give you an exact DNA part. So in case of the non-homologous recombination, the the frequency of error can be a bit higher than the homologous recombination. NHEJ is the DNA repair mechanism that involves direct ligation of the broken ends and that thus does not require a homologous template strand. Okay. And transposones are basically called as the jumping genes. Okay. And transposition is the process of getting those jumping genes involved wherever they get attached to. Okay. CSSR, VDJ recombination and the phenomena of transposition also do not require extensive sequence similarity between strands of DNA involved in the recombination process. Okay, so first the site specific recombination, SSR, also called as the conservative site specific recombination, is a recombination between two defined sequence elements of the DNA. This process is carried out by proteins known as recombinase, which brings together specific ends of DNA, forming a synaptic complex resolving into one of the three outcomes, either insertion, inversion, or deletion. Insertion means some part of the DNA will be inserted at some random place. It's not something predefined. Inversion means the inverted part of chromosome will be added and that will again be of no use. And deletion, sometimes some part of the DNA may be lost in this kind of recombination. Okay. Okay, the types of recombinases in this CSSR are, just a moment guys. Based on amino acid sequences, homology, and mechanisms followed, most recombinases can be classified into two types. They can be serine recombinases or tyrosine recombinases. So, serine combinases cleaves all four strands involved in exchange, while in case of tyrosine recombinases, one strand in each site is cleaved, leading to the formation of a holiday junction. Okay. Recognition site in case of sorry, in case of the tyrosine recombinase is Cre site. Sorry, the recombinase is called as Cre. Recognition site is Lox site, and biological implication is circularization of phage P1. ठीक है. ऐसे ही इन्होंने अलग अलग तरह के जो हमारे पास टायरोसिन रिकॉम्बिनेज और सीरीन रिकॉम्बिनेजेस हैं उनकी इंफॉर्मेशन दी हुई है कि व्हाट आर द रिकॉग्निशन साइट एंड व्हाट काइंड ऑफ अ बायोलॉजिकल इम्प्लीकेशन इज देयर फॉर दीज काइंड ऑफ रिकॉम्बिनेजेस. ठीक है. Mechanism of serine recombinases. One molecule of recombinase catalyzes the cleavage of a single strand. Thus, a total of four molecules of recombinase is required for the recombination process because serine recombinases cuts all of the four strands. Okay. Free hydroxyl of DNA at the 3' and acts as a nucleophile to attack the recombinase DNA complex at the 5' end to generate free recombinase and recombined DNA. Slippery hydrophobic part of the top and bottom halves of the recombinase dimer rotate by 180 degree, and then electrostatic interaction stabilizes initial and rotated state of the recombinase. So this is the process how recombinase is involved. High how interaction happens, because it makes a cut on all of the four strands. Okay, and this is how, by some kind of inversion or something, it makes the recombination, the non homologous recombination. Okay, same goes for tyrosine recombinase. Here only single strand is affected. Sorry. So recombinase cleaves on strand at each site of the recombination. So here only one molecule of tyrosine recombinase is required, while in case of serine it was four. Okay, cut strand is exchange between DNA molecules, formation of a holiday junction happens, and uh, then the finally the strands will be cutted and the exchange will happen. ठीक है, बायोलॉजिकल फंक्शन क्या है सीएसएसआर का? डीएनए रिपेयर में काम आता है. इट अलाउज प्रिसाइज कट एंड रिप्लेसमेंट ऑफ द डैमेज डीएनए स्ट्रैंड, मेंटेनिंग द genomic integrity in E. coli. Farge Lambda uses this method to switch between lytic and lysogenic cycle. Genomic rearrangement CSSR causes gene rearrangement leading to the various recombination, like various combination, like either it can be inversion, it can be deletion, it can be insertion. Bacteria often uses the horizontal gene transfer method to achieve new traits from the other bacteria. Conjugation, that we generally listen in case of the bacteria, either conjugation or pili formation and all. So these kind of things for this horizontal gene transfer, this method is useful. And the development, many organism uses site specific recombination method to regulate tissue specific gene expression and cell differentiation during development. Okay, and it helps in the tracking of the cell lineages during development of Drosophila. CSSR is widely used to manipulate and engineer genomes of organisms, like ablation of any gene function, induction of the gene expression at specific time during development. So for these kind of processes, CSSR is preferred. ठीक है. Next comes the VDJ recombination. So it is generally seen in case of the antibody. Now, one thing, if you guys can relate to me, I'll just tell you if you guys can understand. So basically, when we when our body come across any organism, obviously it's not like it recognizes only single portion of the organism. It recognizes many portions of the organism, so that it can be it can't evade the immune response from the host organism. So for that, it needs some kind of flexibility, so that the antibodies are the recognition ability of the antibody can be enhanced. So for that, VDJ recombination is the one which gives antibody that flexibility, that the epitopes can be recognized, many epitopes can be recognized, the polymorphic epitopes can be recognized easily. This VDJ recombination is very important for antibodies, for our body to produce different types of antibodies for single organism and also for different kind of organisms, so that they can be recognized efficiently, and it won't be evaded if the epitope which is presented to the cells is not the same as earlier. Okay, so epitope is that portion of the parasite or pathogen which is recognized by the antibody. Okay, so it is a specialized set of DNA recombination mechanism that impart enormous diversity in B cells and T cells receptors. This recombination process occurs between specific sites or V, D, and J segments in the genes for the generation of immunoglobulins, that is our antibodies, and TCR, B cell receptors and T cell receptors basically. And these recombination sites are called as the recombination signal sequences. Okay, so there are two types of recombination signal sequences: 12 base pairs and 23 base pair, and recombination cannot occur between the same types of RSS, basically recombination signal sequences. Okay, so these are some mechanism that is developed by the body. Now the NHEJ is a mechanism of repairing double stranded breaks in DNA. Unlike the homology directed repair, which requires a homologous template, NHEJ directly ligates the broken ends without the need of a template. This makes NHEJ an efficient process that can operate in both dividing and non-dividing cells. So it will just ligate the broken ends. It won't replicate. It won't add on something. It will just join the two broken ends without the requirement of any kind of template. ठीक है? And the term non-homologous end joining was introduced by Murray and Haber in 1996. It is often guided by the short homologous DNA sequences known as microhomologies. These sequences are typically found in single stranded overhangs at the ends of double stranded breaks. ठीक है? So, when the overhangs are compatible, NHEJ can accurately repair the break. However, if the overhangs are not compatible, imprecise repair may occur, leading to the loss of nucleotides. So, this is again a very good mechanism, but it requires all of these things, that uh, it should have a compatible overhangs. Okay. Key players are for NHEJ. Key player of this kind of recombination is KU7080 DNA PKCs, Artemis DNA, polymerase, mu or delta, ligase 4, XRCC4 and XLF4. ठीक है. हाँ, so these are the enzymes that are involved with their functions that is defined there. Now the term comes with transposons. As I was telling you, they are also called as the jumping genes. ठीक है. Transposons are the segments of DNA that can move from one locus to a relatively non-specific site in the genome. It is not like they'll go to some specific place, some recognized site. No, it can enter anywhere in your genome. Okay, so movement of transposons can occur without duplication of the element. Accordingly, they are classified as class 1 type or class 2. So there are two classes for transposons, class 1 or class 2. Class 1s are those which can replicate by themselves and can get copy and pasted. Class 2 cannot replicate, they just cut and paste type. ओके कि कहीं से कटे और दूसरी जगह आके लग गए, बट इन केस ऑफ क्लास 1 दे कैन रेप्लिकेट, एंड आफ्टर रेप्लिकेशन दे विल एड ऑन एट सम अदर प्लेस आल्सो. ठीक है. सो डीएनए ट्रांसपोजेजेस कैरी इनवर्टेड रिपीट सीक्वेंसेस एट देयर एंड्स एज अ recombination sites flanking a recombinase proteins called as the transposase, which brings about the process of recombination. So they require a specific protein for this transposons to get fit in, is the DNA transposases. ठीक है. Retrotransposons contain LTR and genes for integrase and reverse transcriptase activity. ठीक है. So outcome of transposition is basically the altered gene expression through insertion within gene. For instance, the IS1 element can insert itself near 80 rich promoter sequences. IS element can cause polar mutations. Rec dependent recombination, site specific recombination between transposable elements in the same molecule can cause rearrangement such as deletions or inversions. ठीक है. तो because of transpositions, because they get they take some piece from somewhere and they can get inserted somewhere else, they can cause a deletion in the portion where they got added to earlier. Okay, so is1 element can cause deletion. TN3 and Y delta resolvase can cause resolvase reaction through deletion. Mutation in general through transposition is insertion. ठीक है. So drawback of traditional methods that has been used in genome editing is they lack precision, the efficiency is low, they are time consuming, and the scope for editing is limited, and obviously then off target effects can also be seen in them. ठीक है. So the other methods for the genome editing can be: genome editing involves altering an organism genetic code using various tools that utilizes the enzymes known as nucleases. These tools follow a common principle. The enzyme is guided to a specific genome target either by guide sequence or by specific DNA binding domains within the nuclease itself. Okay? So after recognition binding of the target DNA, the nucleases can edit the genome through different pathways. If there are double-stranded breaks, then the cell attempts repair through non-homologous end joining or homology-directed repair. But obviously, NHEJ is a bit error-prone method. Okay? So this was just now, how did genome editing came into action? So this is the timeline ki first of all restriction enzymes was there. They used to recognize specific nucleotide patterns and cut the DNA accordingly. That is a method that is being used till now for cloning purpose in our laboratories. Then the zinc finger nucleases, they are composed of engineered nucleases fused to zinc finger DNA binding domain. Third is talens, structurally similar to zinc finger nucleases, but use transcriptional activators like effectors for DNA binding, because these are what they can enter the nucleus, they can reach the nucleus, and they have a DNA binding domain in them also. Okay? So they have a DNA binding domain in them. Next is the CRISPR-Cas9. So this is the genome editing technique in which a guide RNA is involved and Cas9 nuclease activity is there. They can be customized and obviously they can be used accordingly. Then the base editing utilizes the catalytic dead Cas9 fused to DNA deaminases for single nucleotide substitution. ठीक है. Sixth is the primer editing and then the paste, the programmable addition via site specific targeting elements. Okay, so this is how we have reached till here. We started with the restriction enzymes. What are the zinc finger proteins now? So a zinc finger is a compact protein structural motif characterized by the binding of one or more zinc ions contributing to the stabilization of its folded structure. The term was initially coined based on the finger-like appearance observed in the presumably structures of transcription factor 3A. However, it has been identified in a diverse array of protein structure within the eukaryotic cells. So, the discovery of zinc's presence in Xenopus laevis transcription factor 3A, essential for its function, was reported in 1983. तब से जिंक फिंगर जो है कितना चला रहा है. जिंक फिंगर मोटिव्स आर एक्चुअली प्रेजेंट इन E3 ligases also. ठीक है. So it is also present in E3 ligases. Now the zinc finger nucleases. They have again a DNA binding domain and a DNA cleaving domain. So these nucleases are synthetic restriction enzymes created by combining a zinc finger DNA binding domain with a DNA cleaving domain. ठीक है. And the engineered zinc finger domains can be customized to recognize specific target DNA sequences, which allow zinc finger nucleases to pinpoint unique sequences within intricate genomes. So this is how the zinc finger nucleases are, and they can be customized according to the target DNA sequence that we want to target. Alongside CRISPR-Cas9 or TALEN, ZFN या zinc finger nucleases stands out as a significant instrument in the realm of genome editing. ठीक है, these are again the domains, the DNA binding domain or DNA cleaving domain that the zinc finger nucleases has. We are not going into that detail. Then comes the TALEN, the transcriptional activator like effector nucleases. ठीक है. So transcriptional activator like effector nucleases or TALEN are a type of engineered restriction enzyme designed to target specific DNA sequences for the cleavage. ठीक है. They are created by combining a TAL effector DNA binding domain with a DNA cleavage domain which acts as a nuclease capable of cutting DNA strands. ठीक है? So this is the TAL, the transcription activator like effector plus nucleases. So what does that this tails have is basically the nuclear localization sequence, the N terminal translocation sequence, the transcriptional activator domain, and the central repeat domain. So all of these domains or things are present in TAL already. Okay, we are again not going into the detail of this. We have already told you it has a DNA binding domain and it has a DNA cleavage domain also. So application of TAL is this tail or transcription activator like effector nucleases. It is a versatile tool in genome editing. It has been successfully applied to modify plant genomes for improved nutritional qualities and biofuel production. TAL is instrumental in engineering human stem cells and creating knockout models in the various organisms, including cattle, with enhanced tuberculosis resistance. So, these are the applications of TAL. Additionally, it has shown promise in correcting genetic errors causing disease like sickle cell anemia and in harnessing the immune system to combat cancers. It can be a promising way to target these things, and it can be combined with other tools according to your requirements whatever you are doing. Okay, next is the CRISPR Cas system. Everyone must have heard about it. So it is a clustered regulatory interspaced short palindromic repeats, and these systems are sophisticated adaptive immune system employed by prokaryotes. CRISPR is a DNA sequence found in the prokaryotes like bacteria and archaea, originating from previous bacteriophage uh, infections and serves as an adaptive immune system, allowing cells to detect and eliminate similar DNA threats. So basically, here we are. Uh, here we are actually working in uh, boosting the immune response. It is actually found in prokaryotes as their preventive measure, but now it is being exploited in eukaryotes also for our any on our own benefits. So CRISPR-Cas associated proteins or Cas is an enzyme that causes CRISPR sequences as a guide to recognize and open up the specific strands of DNA that are complementary to the CRISPR sequence. ठीक है. General information is the adaptation, the expression and maturation and the interference that will happen in case of the CRISPR-Cas9. So CRISPR-Cas9 system, particularly the type 2A system, is widely studied. Cas9, a 160 kilodalton endonuclease, is the sole protein indeed of the interference. It works in tandem with single guide RNA. So here the guide RNA is involved in case of the CRISPR-Cas9. Fusion of cRNA and tracks rRNA along with Cas1, Cas2 and Cas3 and RNase 3 for DNA acquisition and processing of pre-CR RNA to mature single strand RNA, the guide RNA. ठीक है. So this is again we are not going into the detail of this CRISPR-Cas technique. Okay, we are not discussing it in detail. It has the applications and limitations of CRISPR. So CRISPR's technology has diverse applications impacting agriculture, medicine, and genetic research. Nowadays, CRISPR-Cas is being used worldwide for many kind of processes, for gene knockout, for to study the effect of a particular gene. In research, it is being used intensively. In agriculture, it enhances the crop yield, quality, and disease resistance. In medicines, CRISPR is employed for gene therapy in treating disorders like hemophilia. Hemophilia is a disease in which some of the blood clotting factors are absent, so the person affected will not be, the blood will be clotted in so much time that by the time the blood will be clotted, the person will lose many of a large amount of its blood. Neurological conditions, blindness, and cardiovascular diseases. Additionally, it shows promise in HIV treatment and addressing the genetic disorders. So, the technology has revolutionized genetic editing, enabling precision in modifying DNA sequences. Despite its vast potential, challenges include either concerns, immune responses, and delivery method for clinical applications. So, ongoing research should aim on that part of the CRISPR Cas. Okay? So, this was all about that. You guys must have listened in the last week's lecture. Now, we'll come to the questions in the assignment. Again, I'll ask the questions. You guys will write the answers in the chat box, and no one will speak out the answer so everyone can participate. Okay? So yes, in the REC-F pathway homologous recombination, bacteria uses a repair mechanism specifically for blank in DNA. बताइए REC-F pathway की बात हो रही है. Just a moment, guys. Everyone answer question number 1. In the REC-F pathway of homologous recombination, bacteria uses a repair mechanism specifically for. So sorry, guys. There was some network issue. Are my slides visible to you now? Am I audible and visible to you all? Okay, okay. ठीक है. So question number 1 का answer everyone. REC-F pathway is generally used for. Yes, it is for single stranded gaps, but it can also go for double stranded breaks if the REC BCD pathway is mutated or inactivated. ठीक है. Okay, question number 2. In tyrosine recombinase, just a minute. In tyrosine recombinase, one strand in each site is cleaved, leading to the formation of a blank. Yes, it is a holiday junction that it will form when the cleavage will happen on one strand of each of the strand. Okay. Next, this recombination process occurs between specific sites on V, D, and J segments in the gene for the generation of what? The recombination process occurs between specific sites on V, D, J recombination segments in the genes for the generation of. यस, इम्यूनोग्लोबुलिन्स एंड टी सेल रिसेप्टर्स, या हम इसको कह सकते हैं बी सेल रिसेप्टर्स एंड टी सेल रिसेप्टर्स. ठीक है. नेक्स्ट क्वेश्चन. NHEJ is a mechanism for repairing blank in DNA. बताइए सब लोग. NHEJ किस लिए होता है? Yes, it is for double strand breaks. It is for double strand breaks. ठीक है? Next question is transposons are segments of DNA that can move from one locus to relatively non-specific site in genome known as what? क्या दूसरा नाम बताया था ट्रांसपोजोन्स का? Yes, these are called as the jumping genes. ठीक है? These are jumping genes. ठीक है? In uh, MMEJ end sorry, just a moment, guys. MMEJ end resection reveals short microhomologies on either side of the break, guiding the dash. Guiding what? Guiding what? Question number 6 ka everyone. Guiding the repair process. Yes, guiding the repair process. ठीक है? Next question. The folding of the zinc finger relies on the tetrahedral coordination of the zinc ion with two invariant pairs of. This is a very good question. This is, if you guys will know about E3 ligases. So, on the basis of how many, yes, cysteines and histidines is the answer, right? And how many cysteines and histidines are linked with this zinc ions is the one which defines the different type of E3 ligases. So it is very important here that how many cysteines and histidines are linked with this zinc ions, this zinc finger motif. Okay. The cysteine to histidine to zinc finger motif consist of what? Question number 8, everyone. Question number 8. The answer to question number 8 is an alpha helix and an anti-parallel beta sheet. Okay. So the answer to question number 8 is an alpha helix and an anti-parallel beta sheet. ठीक है? Next question. So the zinc finger nucleases are synthetic restriction enzyme created by combining a zinc finger DNA binding domain with what? Everyone? एक minute. Question number 9, everyone. Answer to question number 9. A DNA cleaving domain. Question number 9. क्या हो गया नेटवर्क को? So sorry everyone, so network issue was there. Question number 9. So zinc finger nucleases are synthetic restriction enzymes created by combining a zinc finger DNA binding domain with. Yes, a DNA cleaving domain. ठीक है? a DNA cleaving domain. नेक्स्ट क्वेश्चन देखा जाए. Question number 10. Zinc finger nucleases use the non-specific cleavage domain generally derived from type 2 restriction enzymes like FO1, which requires dash for DNA cleavage. What does it requires? Question number 10. Yes, it requires what? Dimerization. It requires it to be dimerized. Okay. Question number 11. Talons are created by combining a tal effector DNA binding domain with a dash which acts as a nuclease capable of cutting DNA strands. Question number 11, everyone. What is the answer to question number 11? Yes, it is a DNA cleavage domain. It is a DNA cleavage domain. Yes. Next is the CRISPR consists of conserved repeated sequences interspersed with. Question number 12. Yes, the short spacer sequences. It is the short spacer sequences. Okay. Next question. Just a moment. Someone's there. Someone's there. Let it be. Okay. Next question is in the adaptation stage, the dash complex acquires. The dash complex acquires a protospacer from invading viral DNA and integrates it as a new spacer into the CRISPR array. What is that? Which complex is that, everyone? Question number 13. It is the Cas1 and Cas2. ठीक है? Cas1 and Cas2 complex is this. ठीक है? Next question. In the interference stage, the dash complex identifies the invading target DNA through complementary base pairing. Question number 14. Answer, everyone. Yes, it is B. ठीक है? The answer is B. That is CRRNA and Cas-RNP. ठीक है? Last question. The Cas12a gene codes for an endonuclease that uses a dash to find and cleave viral DNA. Question number 15. Which type of RNA is involved here is? It is the guide RNA. Yes, it is the guide RNA which is involved. ठीक है. To the guide RNA is used here is to find the specific sequence which needs to be cleaved. So it is the guide RNA which through complementarity will tell you that this is the sequence that needs to be cleaved, that needs the CRISPR-Cas action. Okay. So with this, the questions of the assignments are over. If you guys have any more queries, you can ask me for the week 11 and for the previous week if any. Next week will be our last week's discussion. I hope you guys understood the course, you guys understood the things that was taught in the course. Still, if you have some kind of confusion, some questions, you can ask me. और आ जाए दिल्ली के नेटवर्क. TTC. Just a moment, everyone. There's some connectivity issue here. Hi, sorry everyone, there was some connectivity issue here. Mm. हाँ जी, तो किसी का कोई क्वेश्चन कुछ कोई पूछना चाहता है, so they can ask me, otherwise you guys can leave the meeting. Thank you so much for joining. Okay, bye class. Thank you so much for joining. Mhm. Stop क्यों नहीं हो रही है?